And today we are going to talk about and discuss about cell membrane or plasma membrane. In my previous two videos, we have discussed about cell organelles, and this time from the same chapter, cell, that is the fundamental unit of life, we are going to take up the topic cell membrane or plasma membrane. Now this plasma membrane is a unique structure which is actually making your cell a unit of life, fundamental unit of life, because this plasma membrane is responsible for separating the interior of cell from outside environment, right? So it is basically making a covering outside your cell in which all your small entities are going to be kept, right? So this covering, which is actually protecting the cell from its outside environment, is known as plasma membrane or cell membrane. These are two different names. One more name was earlier used to be used. It was plasma lemma. So that is also one another name for this plasma membrane. All right. Now let us see the structure of plasma membrane. The scientist who gave us this structure was Singer and Nicholson, and that is why this is still called as fluid mosaic model. This was the name provided by them. These scientists provided this name, fluid mosaic model, and this name is still used. So this is the fluid mosaic model of your plasma membrane. This is basically very much enlarged structure of your plasma membrane, which generally seems like a thin line, right? Now let us see what this structure is actually made up of. First thing you need to see is or notice is this particular term, lipid bilayer. This is actually lipid bilayer. So where is that bilayer? Can you see this? If I'll draw a line here, this entity and this entity is exactly similar to each other. So this is one layer and this is another layer, right? This is why it is bilayer. This fluid mosaic model was also used to call as sandwich model. Why is it so? Now dig in deep into this layer. You can see this circular structure which are having two tail-like structure. These are each entity, right? Repetitive entities. Now this circular head-like structure is nothing else but your phosphate. And these tail-like structure are nothing else but lipid. So this one molecule is basically combination of phosphate and lipid, and that is why we call it phospholipid as well. Can you see it written here? Phospholipid. So this phospholipid is actually going to make one layer, and just like that another layer. Now consider if this protein to be like a bread for your sandwich. So you have got two bread at the top, at the bottom, and in between the lipid tails are filling up, right? So it will, if you can consider this lipid as jelly or jam, then this becomes your sandwich model, right? Two bread and in between jam, right? So this fluid mosaic model is also called as sandwich model. Now we can see these big boulder like structure which are actually invading my plasma membrane. What are these? These are protein molecules, right? Certain molecules you can see which are completely entirely breaking the plasma membrane. That means they have connection with the outside of cell and also connection with the inside of cell, right? So they can actually serve as channel, transportation channel for various things to come inside or go outside, right? So these proteins are there for a purpose, and that purpose is transport across the plasma membrane. There are certain smaller protein also which gives them specific strength and further various quality depending upon where they are present. Alright. So let us see what our plasma membrane has got specific in it, and that specific thing is selective permeability, right? This is the most important thing which your plasma membrane or cell membrane has, and this selective word means, but very obvious, it's clear from this word, it is going to select what all materials can come in and what all can go out. Basically it is going to allow the entry and exit to only certain selected materials. Not everything can cross your cell membrane. right? So now in this selection, few selection are always welcome, like certain solutes cross membrane freely, while some can cross with assistance and others cannot cross at all, right? So there are three category of different type of solute and solvent which can cross the cell membrane. Now useful molecules can enter the cell. This is the very prime rule. Whatever is required can come in. Metabolic intermediates remain in the cell. Like your cell is actually doing the glycolysis, or digesting out or breaking down your glucose. So in between, if it is making pyruvate and that pyruvate is going out of the cell, in that case the complete glycolysis will be disrupted. So that's why metabolic intermediate needs to be there, held inside the cell, so that the complete process can happen. So this is one thing. Another thing is various type of secretion and waste can easily leave the cell. So everything is depending upon the requirement. And the last and most important, overall cell has to maintain the homeostasis. That is a particular condition of normal. That can be different for each cell, right? Each different type of cell. So homeostasis can be different for different type of animals, different type of cells, right? So it has to maintain a normal, and so hence it has to maintain homeostasis. Now we'll talk about what things can draw inside a cell and what can go outside the cell, right? So very first thing is, of course, we want certain metabolic processes to happen inside the cell. So we want all the raw material for the different type of metabolism to enter the cell. Like we want food to come in so that it can be broken down and digested. We always want water to come in. Rather, water is the only substance which has got free movement across the cell membrane, whatever type of cell it is. It doesn't matter. Water can move always throughout the across the cell membrane. Various type of cell, animal cell, plant cell, various different organ cell, whatever. Water has got free passage. Now we require at times certain type of salt to enter, to so that certain metabolism, if a salt is going to assist in certain metabolism, we want that salt to enter. So that salt will also enter. Oxygen also, we always want oxygen to come inside, because this will be going to generate the ATP, which will be fuel for my various type of processes, right? So oxygen is required, water is required, so they are always welcome. Now product of metabolism, the second thing is regulatory substances. Now regulatory substances like various type of vitamins and hormones which are required by that particular cell, those also can enter the cell. They all can come inside a cell. Alright, now we'll talk about substance which need to be out, which need to go out of the cell. These can be products of metabolism, like all those metabolism which was happening here will result into a particular product and byproduct. All those waste which are created by metabolism need to go out, like nitrogenous waste, your carbon dioxide, they all need to go out. Okay, so they are free to go. Now various kind of secretion, waste protein, enzyme, hormones, milk, tear, mucus, antibodies, these are various type of secretion which your cell makes. So when cell is producing them, they are supposed to go to certain area so that they can be effective. So those also can easily leave the cell. Now let's see the transport of various material across the cell. We have discussed that it is going to select the transport, but exactly how we can categorize the transport across the cell? We can categorize into two types. Okay, now one type is your passive transport. Passive transport means when you are not using energy, any kind of energy. So energy is not going to be used. And the active type of transportation is when you are going to utilize the energy. Alright, so passive transport, we have diffusion in it and osmosis, which is very much in detail for your syllabus. We are going to discuss it in real detail. And then active transport, where we are going to utilize the energy, like endocytosis and exocytosis. We will be discussing these two as well. So very first type was diffusion. Now What is diffusion? Diffusion is, by definition, the diffusion is spontaneous, random movement of molecules from a region of their higher concentration to a region of lower concentration. So where we are going? We are going from higher to lower. Why we are calling it spontaneous movement? Because in the previous slide I told you this is a passive movement. That means it does not require energy, so it is going to happen by itself. So it is a spontaneous movement. Why it is random movement? Because there is not necessarily a plasma membrane going to be there in between the two different things while diffusion is happening. It is not necessary to a plasma membrane to be there in order to diffusion to happen. So that is why it is random and spontaneous. Rule is only one, that it is going to happen from a region of higher concentration towards a region of lower concentration. So if I'll discuss that there is salt, which is 10 gram of salt is dissolved in one solution. So, and if 2 gram of salt is dissolved in another solution, so one solution is going to have 10 gram of salt which is concentrated, while other one is 2 grams, so it is less concentrated. So salt will simply start moving from 10 gram to 2 gram till the time equilibrium is reached. So this is basically for making the equilibrium reach. Now examples can be movement of CO2 and O2 and absorption of nutrient by your intestinal cells, right? These are various examples for diffusion. Now let us see the diffusion. In the first picture you can see around 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10 molecules on A side, right? And on B side you can see only one molecule. So that means there is a concentration gradient which has been created. This side has got lower concentration while this side has got higher concentration. Since the plasma membrane is not very much required, solute can freely move, it's a random movement. So what will happen? The movement of particle is going to happen from A towards B, and it will keep on happening till the time you'll see almost equal solute on both sides. Now you can see here and here, both sides you can see equal number of solute. So what is happening? From higher concentration to lower concentration, a movement is happening which is leading to equalize or normalize the concentration, right? So till the time the equal concentration is achieved, till that time the simple diffusion will keep on happening, right? This is one rule of diffusion. Now the common thing which we have already discussed, that diffusion happens for oxygen and carbon dioxide when we talk about your cell. And because cell is always having plasma membrane or cell membrane covering it, so this diffusion is also going to happen across the cell membrane. But since oxygen and carbon dioxide are very, very small molecule, so they are freely movable inside and outside. So what is going to govern this movement? Only the concentration, right? So we can see oxygen is higher on outside, right? And high concentration of CO2 is inside. So oxygen will start moving inside while carbon dioxide is going to move outside. Now I believe that diffusion is clear. So we'll go ahead and study osmosis. Now osmosis is not very different than your simple diffusion. Rather, it is also a special case of diffusion only. There are few differences and we are going to discuss them in pretty detail. Okay. Now since this is a little confusing topic, I am going little overboard and I am explaining it in detail for better clarity. Alright. First thing we need to notice is diffusion is about water. When we talk about diffusion, about water through a partially permeable membrane, or simply in simple word we can call it our cell membrane. Then we are actually talking about osmosis. So osmosis is nothing else but diffusion through semi-permeable membrane. Like in simple diffusion, presence of a semi-permeable membrane was not necessary. But for osmosis, this become necessary. This is first point. And second point is, in diffusion, when we were looking at the equilibrium reach, then in that case solute particle were moving. Here we are talking about movement of water molecule. That means we are talking about movement of solvent. So these are two basic differences between diffusion and osmosis. Rather, I can say these two particular things are making osmosis a special type of diffusion. Alright, one thing is a permeable membrane, partially permeable membrane, that is your plasma membrane or cell membrane, is at most necessary for osmosis. And second is this time only solvent molecule, and in in that solvent also specifically water molecule movement is only which we are going to notice. All right, now osmosis always happen in comparison. Okay, so now if we'll see two solutions which are having different concentration are going to be separated by semi-permeable membrane. In such cases we will see that diffusion of water happens on the basis of the concentration gradient, right? I'll explain you this point. And next thing is movement of solvent water from a region of its higher concentration to lower concentration. Now in this line is common, higher concentration to lower concentration, right? In diffusion also we were noticing this, but in that case what was like which concentration we were talking about? We were talking about concentration of solute, right? So if a particular solution is concentrated, then solute molecule will start moving from the concentrated solution towards the dilute solution. But here we are going to watch the movement of water from region of its higher concentration. So it's become, this particular word become the most important thing. Water is definitely going to move, and on the basis of water's higher concentration to lower concentration. What does it mean? I'll explain you better in the next slide. Just look at certain examples. Examples are absorption of water from soil by root of plants. This is one example of osmosis. Second example can be reabsorption of water in kidneys. Alright. Now let us see the difference between diffusion and osmosis. Alright. This is diffusion. We can see if there is a semi-permeable membrane, because we are talking about all kind of movement across cell. So we are going to keep in consideration that semi-permeable membrane or cell membrane is always going to be there. Okay, so if a particular solution, and these blue dots are representing your solute particle, while this blue sky blue solution is actually representing your solvent. Okay, so if two different type of solutions are separated through a semi-permeable membrane and one solution is clearly concentrated, we can see more blue dots. Rather here on the second part there is no blue dots, right? So what will happen in diffusion? These particles will start moving from here to here, that is from higher concentration to lower concentration. Till what time they will keep on moving? Till the time the equilibrium is reached. That means the solute particles are almost equally distributed in both the sides. This is simple diffusion. Alright. Now look at the situation when we are talking about osmosis. Now again we are going to separate two different solutions by the help of a semi-permeable membrane or a cell membrane, right? Now we have these blue dots, we have actually shown you the water molecule with this blue dots, right? These are water molecule, H2O. Why we have shown you the blue dots for the water molecule? So that you will easily understand the diffusion, the movement of those molecules, right? You can see blue color is H2O while red color dots are solute particle. So say in one solution there is pure water, okay, so only we can see blue dots, that means H2O particles, while second one is a lower H2O concentration and higher solute concentration. We can see the comparison. If we'll see the comparative number of H2O molecule, they will be comparatively less because certain space which could have been taken up by the water molecule has been taken up by these solute particle, right? So here this is a solution in which water and solute particles are mixed, right? So here if we'll say there is going to be more water concentration in comparison to second part which is having comparatively lesser water concentration, because certain place where water can be is now taken up by your solute particle. Okay, so now what was the rule? Movement from higher concentration to lower concentration, right? Here also we saw higher concentration to lower, but this time here which particular concentration we are talking about? We are talking about concentration of H2O. not the solute. Here we were talking about solute but not in the osmosis. So water concentration is here higher, while in this case it is lower. So water will move from its higher concentration to its lower concentration, right? So water will start moving from pure water to your solution, right? It will keep on moving till the equilibrium is reached. Now see, at any case, in the second picture you can see the solute particle is still stuck into the second part of the beaker. They are not here in the first part. So how equilibrium is reached? Equilibrium is reached on the basis of how many water molecules are here on the first part and how many water molecules are there in the second part, right? Because certain space in this volume has been taken up by the solute particle, there can be lesser water, and this is pure, so all water will be there. So what will happen? The water will keep on going this way till the time the number of water molecule in first place is equal to number of water molecule in second place, which will result in a volume difference, right? Now if we'll count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 14, 13. Roughly 13 particles are here. So roughly 13 particles should be here. And since this is a solution, so volume is going to definitely increase, because in the same level, same volume, out of 13 for sure 3 or 4 would be your solute particle and 9 or 10 would have been your water particles, right? So in order to make number of water molecules equal, this movement will start happening. So basically what is happening? Water is moving from its higher concentration to its lower concentration. Alright, now I'm going to go ahead and explain you in terms of cell. Before that, we see the difference, because this difference also comes at times. What could be the difference between diffusion and osmosis? The very first difference is semi-permeable membrane is not required. This is not necessarily required. We are seeing all the experiment with the semi-permeable membrane because we are discussing the movement across cell, and that is why the semi-permeable membrane is there, but they are not necessarily required for simple diffusion. Simple diffusion can be taken in account as, you know, in a room, in a corner, when you are spraying a deo, from that corner it will spread into whole room, right? There is no semi-permeable membrane in your room, right? And the this particular mixing of, you know, the movement of the concentrated fragrance particle, the deo particles which moved and became equal in the whole room, is simple diffusion. Okay, so here also semi-permeable membrane is not necessarily required. But when we talk about osmosis, semi-permeable membrane is required. This is a necessity, this is must. This is first difference. Second is solutes are going to move across concentration gradient. While in osmosis, solvent molecule move across a concentration gradient. This is the second most important difference, right? Here the solute particles are moving easily, while here only the solvent, and that too specifically water, is going to move. Okay, this can take place in any medium. I have already told you an example where the deo particle were moving. It was in gaseous medium, right, in the air, but this osmosis can take place only in liquid, right? This is definitely going to lead to equalization of concentration gradient, right? While in osmosis does not necessarily equalize the concentration gradient. We have seen in the previous slide, this will only equalize the concentration or the number of molecules of water in two different situations, not the concentration gradient. All right. Now let us see in terms of solution and your cell. Okay, there are three types of solution. One is isotonic solution, second is hypotonic, and third is hypertonic. All these three types of solution are comparative name. Okay, no single solution can be hypotonic, no single solution can be hypertonic. It is always in comparison. So these terms can be used only when you are comparing two different liquid. What liquid we are comparing here is, we are comparing the liquid present inside the cell and liquid which is outside the cell, because we are talking about cellular transportation. All right. So isotonic means, iso means same. Okay, so same, that means inside the cell the concentration is similar to outside the cell. Okay, so sorry, I was looking at a wrong picture. This is the isotonic one, and we can see that the concentration inside the cell is equal to concentration outside the cell, right? So this, in this situation, cell will remain its own size because it is in same tonicity, and this is isotonic situation. Now what about hypo? Hypo is always less, right? So just remember it like hypo means less, okay? So below or under, that means the tonicity is going to be less. In other word, you can also call this solution as dilute solution, right? If solution is going to be dilute, that means less tonicity, that means less solute. Less solute means more water. It is a vice versa term, right? So less solute, more water. You remember the pure water and solution slide? Should I go back? Here, the pure water and the solution. So pure water was having higher concentration of water, while the solution was having lower concentration of water. Why? Because of the presence of solute. So just like that, here when the water is, or when the solution is dilute, that means it has less solute and more water. It's always vice versa. Okay, so what happens in hypotonic situation? Hypotonic, that means the solute particle are going to be comparatively less in comparison to what is present outside the cell, right? So blue color particle are comparatively less and blue color solute particle are more outside. So what will happen? If blue color particle are more, that means the more water is present. That means in hypotonic solution, like in comparison to cell, right? So in when we are comparing the cell, that means we have kept a cell in a solution where lesser solutes are present outside in comparison to cell. That means more water is present outside in comparison to cell. More water outside, less water inside. What will happen? More water will start flowing inside your cell and cell will swell up. Okay, so this is your hypotonic situation. In this, cell generally swell up. Okay, now the last one is hypertonic. Hyper, just the name suggests, this person is very hyper, the nature. That means he is very exaggerated. He is getting very angry or all those things, right? This is like, the term very is very much attached. So please remember it by that, that overly or more, right? So hyper means more solute, right? More tonicity, that means highly concentrated, and when more solute, then vice versa, and less water. Highly concentrated, less water, more solute, right? So what is happening? Going to be more solute outside, right? A cell is kept in a hypertonic solution. That means the cell is having lesser solute in comparison to the outside world. And that means the water volume is less outside and more inside. So more water will start moving towards the less water, right? Higher to lower. So water is going to come out of your cell. If water is going to come out of your cell will become shrunken, right? So cell will shrink. Okay, let's discuss this in detail. See, we know that cell contain biological molecules like sugars, salts, and thus comparatively less water content. Definitely we are sure that cell does not have pure water only inside it, right? So when cell is actually having a solution in it, right? Because if there is any sugar or salt, it will get mixed in the water content what is present inside the cell and it will become a solution. So cell is always containing a particular solution which will have a particular tonicity, okay? So water may move in or out on the cell depending upon the concentration which we have already discussed, right? Now see the isotonic again in detail. So what is happening in isotonic solution? The solute is, the concentration of solute is equal on both sides, as well as the concentration of water is also equal on both sides, right? Water is also equal in both sides. So in that case, water will come inside, of course, because cell membrane is always permeable for water, and water will keep on going outside as well. So what is going to happen? The water movement is going to be there. Equal amount of water is going to come in and equal amount of water will go out. That will result in no net movement and cell remain of same size like normal. Okay, so basically water molecule are definitely going to according to concentration gradient. Since there is no concentration gradient created and they are free to move across the cell, so same amount of water is going to come in and same amount of water is going to go out, leading your cell to stay normal in normal situation. Okay, now when we talk about hypotonic in detail, we have already discussed hypo means less. That means your cell is kept in a hypotonic solution. Now tell me where the water will be more, inside the cell or outside the cell? A hypotonic solution means less solute, right? Less solute means more water, right? And less solute means more water, and more water is going to lead to formation of gradient. So inside the cell there is comparatively lesser water and outside there is more water. So water will start going inside your cell, right? So cell is actually kept in a solution where the concentration is comparatively less. That means more water, less solute, and that will result into movement of water inside. See again here, the movement of water outside will also happen. It's not like that only inside movement will happen, because water is free to move across the cell. Movement of water will happen in both the direction, but more water will come in and less will go out. You can see three arrows here, two I made, three arrows which are pointing towards inside and one which is pointing outside. So basically more water is going to come in and less water is going to go out, resulting into net water collection inside the cell, right? And since the cell is getting water, it will swell up, right? Become turgid, right? So water will move inside the cell as the water potential is higher outside than the inside, right? Now here is a very interesting fact. When we talk about animal cell, we know that animal cell has got cell membrane, and outside to cell membrane there is nothing protective, right? So if an animal cell is kept in a pure water, like beaker in having pure water, then water will continuously keep on coming in, resulting into swelling. Now cell will keep on swelling and at a point it can burst, right? But this will not happen when we talk about plant cell. Can anyone tell me why? Yes, you are absolutely right, because of the presence of this thick, rigid cell wall. So cell wall is not going to let your cell burst. Though the cell will become turgid, very much swelled up, but cell wall will not let this to burst out. Okay, so the cell will only be turgid, but animal cell can burst. Now talk about hypertonic. We have already discussed hyper means having large number of solute, and obviously vice versa, less number of water, less quantity of water in it, right? So if a cell is kept in a hypertonic solution, so the solution definitely is going to have more solute, less water in comparison to cell. So what will happen? This is the first stage, okay? And this is second stage. So first let's talk about first stage. What happens? The cell is kept in a hypertonic solution. There is more solute outside, less water inside, less water outside, and more water inside. So what will happen? Again, both direction movement is going to happen, but more water will come outside in comparison to lesser water coming inside. So what is happening? Three arrows are taking water out while one arrow is bringing water in. So cell is going to ultimately lose water. More water will go out and less water will come in, leading to shrinkage of cell. Now the shrinkage of cell happens in certain stages. First, when the cell becomes little flaccid, that means cell is shrinking, right? Not turgid anymore. Then your cell, if the cell is kept in a very high concentrated solution, in that case water will keep on going out, right? It will not stop. So this, what will happen, the cell wall, right, the plant cell, especially in plant cell we see this phenomena, the cell wall and cell membrane are always in close connection, but because water is getting lost continuously, what will happen, the cell membrane is going to leave cell wall, and all the cell content is going to come in a specific place, like at a point, at a corner, it will get collected. In such situation we call it plasmolyzed cell. Now the cell is so much dried up, shrinked, that we call it plasmolyzed cell. In plasmolyzed cell also there is a situation when cell cannot revert back, right? So that situation is called as incipient plasmolysis. Incipient plasmolysis, when this stage is reached, in such case, when the cell is kept back into fresh water, it does not take up water anymore. That means cell dies. Okay. So hypertonic situation, more water is going to go out in comparison to whatever is coming in, right? So water will move out of the cell. Okay? And because water potential is higher inside this time, water is more inside, so potential is higher inside in comparison to outside. That is why water is moving out. Okay, and cell is going to shrink, cell will become flaccid first, and then plasmolysis, and then it will be a plasmolyzed cell. I hope this is clear to each and everyone. So we have discussed the osmosis, and diffusion and osmosis are two different parts which comes under passive transportation. Passive, again I'm telling you, passive means the transportation where no energy is required. So let us talk about active transportation now, where you will be requiring energy for the transportation to happen. And the very first example is endocytosis. Yes, by the picture, by the diagram, you can easily make out that endocytosis is one process which you can easily see in amoeba, right? So what is endocytosis? Endo means inside. Cytosis, inside the cell. Cyto is for cell, endo is inside. So endocytosis, or another term is phagocytosis, is the movement of something from outside to inside, and phagocytosis generally is for something solid. Okay, so some solid substance is going to move from outside to inside. How it happens? We can see the example of amoeba. Amoeba, whensoever it detects any food particle, say this is food particle for amoeba, so what will happen, the amoeba is going to start moving its pseudopodia. These two things are its pseudopodia, right? It start growing a pseudopodia till the time it is reaching towards the food, and when it is in contact with the food, it will grow quickly its pseudopodia, so that both the pseudopodia touches each other, right? And once they touch each other, can you see this black color line? The moment these two pseudopodia meet, right? Here this tiny cell membrane will dissolve and these two will fuse, resulting into formation of a food vacuole, right? So now what is going to happen? Through endocytosis, some food particle came inside. This requires energy. This is one type of process. In your cell also certain endocytosis happen, right? Now the next thing is exocytosis. In the same thing, in the same energy expenditure process, exocytosis, the name itself suggests, exo means outside, cyto means cell. So this time the movement is going to happen outside the cell, like all those enzymes which are getting formed inside your cell, hormone if it is getting formed inside the cell, they need to be released. If they need to be released, this exocytosis is the process for that. How it happens? A simple vacuole or something is going to bring that molecule or solute or particle which need to be going out. They will come and come near to the plasma membrane, and then these two membrane are going to fuse. The moment it is fusing, it can release the particle outside, right? This is cytoplasm, this is exterior. So this is exocytosis. We saw that from inside the cell, the particle went outside the cell. So this is example of exocytosis. This release also require energy, and that is why it is categorized under active transport, right? So exocytic vesicle generally fuses with the plasma membrane, which I have just told you. All right. So this is all about your transport across the plasma membrane. Now do like my video and subscribe, and let me know in the comment section if there was any confusion still left. Then in that case, I'll make another video for sure. And thank you so much for watching. Bye bye for now.